Digestive physiology are the processes of actually breaking down the food. Digestion is simply the breakdown of food so it can be absorbed and used by the body. Mechanical digestion is the grinding, chopping, and mixing of the food, taking the large chunks of food and breaking it down into finer chunks. Chemical digestion involves taking the macromolecules, proteins, lipids, carbohydrates, and breaking them down to their monomers. In order to do this, we're going to have enzymes. For mechanical digestion, the teeth and the tongue begin the grinding and chopping process. We then swallow, deglutition, and peristalsis takes over. Peristalsis are contractions, smooth, regular contractions that propel the food through the gastrointestinal tract. At certain places along the gastrointestinal tract, segmentation occurs. These are contractions that help mix the food with the enzymes. In chemical digestion, we have to break carbohydrates down to monosaccharides. Proteins have to be broken down to amino acids. Lipids have to be broken down to monoglycerides and 2 fatty acids, and nucleic acids have to be broken down to nucleotides. All chemical reactions in the body are mediated by enzymes. You'll remember that enzymes are proteins. They are catalysts that speed up reactions. They can't make a reaction happen that wouldn't normally happen, but they can make it happen thousands and thousands of times faster than it would normally. Because enzymes are proteins, they're subject to denaturation. pH, heat, agitation, and ionic imbalance can all denature proteins. Let's look first at carbohydrate digestion. Polysaccharides are usually taken in primarily as starch. We have amylase in the saliva, and then we have to wait till we get to the duodenum where the pancreas can slip in some amylase to break down polysaccharides. Amylase will eventually break polysaccharides down to disaccharides, primarily maltose. The brush border enzymes of the small intestine will then break the disaccharides down to the monosaccharides, glucose, so that it can be absorbed. In testing for carbohydrate digestion, if we have starch and we add iodine to it, it gives us a dark blue color. If you add your saliva to some starch that should have amylase in it, it should break down the starch to maltose. If we add iodine to maltose, we don't get that color change. We just get the color of iodine. For protein digestion, proteins begin to be broken down by pepsin in the stomach. We get polypeptides. Then proteases from the pancreas will begin to break the polypeptides down to peptides, and eventually the brush border enzymes of the small intestine will break the peptides down to amino acids. One of the ways we can test for digestion is to use gelatin. Gelatin is a solid protein. Meat tenderizer contains papain. This is a protease. It's a protein enzyme. It breaks down protein. As papain breaks down gelatin, the gelatin will liquefy. Mostly our lipids are triglycerides or neutral fats. We really don't break down lipids until we get to the small intestine. We need bile from the gallbladder to emulsify the fats so that we get smaller fat droplets. And then the pancreatic lipases will break the fats down to monoglycerides and 2 fatty acids.